Hi everyone, my name is Miss Berceros and I'm going to go ahead and lecture on cell transport. All right, so the main cell part that we're going to be talking about during this unit is the cell membrane. If you guys can remember from our previous cells unit, the job of the cell membrane is to let things in and out of the cell. So to start, the cell membrane is what we called semi-permeable. Okay, so it is a semi-permeable lipid bilayer. This lipid is another word for fat, and bilayer, bi as in two, so it's made up of two layers of fat with proteins that are kind of spread out within the membrane. So this word right here, semi-permeable. Semi-permeable is this. So the cell membrane only allows for some materials into and out of the cell. So the cell membrane selectively chooses what can enter and exit the cell. It's mostly made up of fat, like I said, and most of the time, the smaller molecules can pass through the membrane pretty easily, and then molecules that are larger are not able to pass through. This is just an example of some things that can pass in and out of the membrane. So on the left, we have things like water, oxygen gas, carbon dioxide. These are things that can pass through the membrane relatively easily. And then on the middle and right-hand side, these are some things that either need a little bit of help to pass through or cannot go through. All right, diffusion. This is going to be an important term for us. So when we talk about diffusion, guys, we're talking about the random movement of molecules through a membrane. And when they move, we're always going to go from high concentration to low concentration. So we're going to move from where there's a lot to where there is a little. Okay, and the best way that we can demo this is if we take a look at our picture on the bottom left here, we can see that we have like two areas separated by this membrane in the middle. On the left, we have lots of molecules. On the right, we have only a few molecules. So diffusion is going to be the molecules on the left moving over to the right through the membrane from high, where there's a lot, to low, where there's less. Diffusion is only going to happen when there's a concentration gradient. So this means when there's a difference in the amount of molecules in two different places. And then diffusion is a type of passive transport. Okay, so when we say passive transport, what we really mean is that we do not require energy for this process to happen. It happens all the time without any energy needed. All right, the goal of diffusion. The goal of diffusion is to reach what's called equilibrium. Okay, so equilibrium is when there's equal concentrations on both sides of the membrane, so there would be no gradient here since both sides are the same. Something important for us to note is that when we reach equilibrium, all of the molecules are still moving. We're still going to have some movement back and forth, except now it's going to be equal back and forth. So the molecules are still moving. On this slide, we just have some examples of diffusion. Best example I like to give of diffusion is if you come home from school and you can smell one of your family members cooking dinner, that's an example of diffusion. If you drink coffee, If you put creamer in your coffee and then you can see the cream move through the coffee, it's also a good example of diffusion. So how does it work, right? If we pretend that this blue circle is our cell, right, we can see that there are more molecules on the outside. So if we're moving, remember, from high to low, our molecules are going to move in to the cell. And then once we reach equilibrium, it will look more like this cell right here. The number of molecules inside and outside of the cell are equal. Diffusion can also look like this, right? So if we have all of, let's say, our sugar molecules at the bottom of our lemonade, eventually all of the sugar will spread out through your entire cup of lemonade and they'll move up. So there are a couple of things that can affect how fast diffusion can happen. The first one is temperature. So hotter materials, so hotter gases, hotter liquids, all of those molecules within those hot either gases or liquids are moving faster. So hotter things move faster. So if this happens, that means diffusion is also going to move faster. So when we have, for example, boiling water, if you can picture a pot of boiling water, the water is moving really, really fast. The water molecules are bouncing all over the place. If we were to introduce like food coloring into that pot of boiling water, the food dye would spread out throughout the pot very, very fast. So faster molecules increase the rate or speed of diffusion. Colder temperatures are going to slow down diffusion. Okay, the other thing that can affect how fast diffusion happens is concentration. So if there's high concentration, which means there are lots of molecules, the more molecules there are, the more they're going to be kind of bumping into each other. All of these collisions are going to make things move faster. And faster molecules, again, are going to increase the rate of diffusion. Osmosis. So osmosis, guys, is a kind of diffusion, but osmosis only refers to water. It has the same goal as diffusion, which is to reach equilibrium. So we want equal concentrations of water on both sides of the membrane when we're talking about osmosis. So just some quick examples of osmosis. If we're talking about noodles being put into a pot of water, that would be an example of osmosis. If we're talking about water coming out of lettuce in a salad, that's another example of osmosis. All of those involve water. So just like in regular diffusion, water is going to move from high concentration to low concentration. If we take a look at this example here, we're going to say that these orange solute molecules cannot move through the membrane. So in order for us to reach equilibrium in this example, all of the water, or not all of the water, some of the water is going to have to move from right to left. That's the only way that we can reach equilibrium here. All right, so let's talk about 3 different kinds of solutions. We're going to do some examples of each of these in a little bit, but let's talk about what they mean. So, first one, an isotonic solution. An isotonic solution is when the concentration of dissolved molecules or solute and water is equal to another. So this solution would already be at equilibrium. If we have an isotonic solution, it's already at equilibrium. So we have equal amounts of water in and out of a cell, for example. Okay, again, just because we're at equilibrium, we still have movement. And then also when we're talking about an isotonic solution, the cell is going to stay the same size. Because again, remember, we're going to have equal amounts going back and forth, so the cell isn't going to change size. All right, our next solution is called a hypotonic solution. In this kind of solution, we're talking about when the concentration of water is higher than another solution. So an example of this, guys, is we have more water outside the cell. Okay, so if that happens, remember we want to move from high to low. So what this means is that the water would move into the cell. Okay, and if we think about it, if water is going to move into the cell, our cell size is going to increase. Okay, and you're probably thinking, how am I going to remember how that a hypotonic solution, the cells increase? The way that I like to remember it is hypo is very similar to hippo, the animal. And if we think of hippos, hippos are really big, so hypotonic solution, hippo. All right, our last kind of solution, guys, is a hypertonic solution. And in this case, we're talking about the concentration of water is lower when compared to another solution. Right? So this example, we're talking about more water on the inside okay, and if this is going to happen, since we move from high to low, that means the water moves out of the cell. Okay, and if our cell is losing water, that means our cell is going to shrink. Okay, so the way that we can think about hypertonic solutions, it's not as catchy as hippo, but hyper has an R and shrink also has an R. That's what I like to think about. So iso, the same or equal, hypo, hippo, our cell gets bigger. Hyper, our cell shrinks. Okay, this is just a graphic of our three different solutions. I'm going to skip past this because we're going to go into each individually. So our first, for an isotonic solution, remember the water concentrations are equal in and out of the cell. Water is going to move equally in and out of the cell, and the way that we can show this is, if we can see in this picture, there are double-sided arrows. This is going to show water movement equally into and out of the cell. There's overall no gain or loss in water, no change in cell size, and remember, these kinds of solutions are already at equilibrium. All right, so if we take a look at our example here, we have a beaker filled with water and some salt, and then inside the beaker we have a cell. All right, so the first thing that we're going to do is we're going to look at the amount of solute. In this case, we're looking at the amount of salt, and we're going to use this to determine how much water is in the solution. All right, so if we think about it, 100% is the highest percentage that we can have. So if inside our beaker there is 10% salt, that means that 100 minus 10, there has to be 90% water. I'm gonna abbreviate that H2O. Okay, so 10% salt inside means that there is 90% water, right? And then if we look inside of our cell, again, it also says that we have 10% salt. So we're also going to have 90% water inside the cell. So clearly this solution is going to be isotonic because our percentages of water are the same. Water is going to move in both directions. And this example is at equilibrium. All right, let's look at the hypotonic solution. So again, when we're talking about a hypotonic solution, there's going to be more water outside the cell, so water is going to mostly flow into the cell. So the characteristics of a hypotonic solution, guys, our cell is going to gain water. Swell means to get bigger. Okay? And then in some cases, if the cell continues to get too big, the cell could burst, and this is called cytolysis. So cyto is cell, lysis means to break. And again, our end goal is always going to be equilibrium. All right, so just like our previous example, we're going to look at the solute to find the amount of water. So inside of our beaker, just in the solution, we have 10% salt still. So if we have 10% salt, that means we're going to have 90% water. And then inside of our cell, now we have 20% salt. So 100 minus 20 is going to be 80% water. And remember, since we go from high to low, there's more water outside, so our water is going to go into our cell, which is going to increase our cell size. All right, let's look at the last example of solution. So in a hypertonic solution, we have less water outside the cell. So water is going to mostly flow out of the cell. If this happens, our cell is going to lose water. If our cell loses water, our cell is going to shrink. And still, our end goal is equilibrium. All right, so let's look at this example. We are going to, again, look at the solute to find out the amount of water. 20% salt will give us 80% water. On the inside, 10% salt, 90% water. So we always move from high to low when we're talking about osmosis. So our water is going to move out of our cell, which means our cell will shrink. All right, so let's do some examples, okay? We're always going to first try and figure out how much water is inside the cell, how much water is outside the cell. Sometimes it will give it to us, sometimes it will not. So if we look at this first example, outside of our cell we have 85% water, inside 90%. Remember, I'm going to move from high to low. So in order for us to show this happening, I'm going to draw arrows showing that water is going to move out of our cell. So if I go ahead and just fill in the blanks here, there is more water inside. So water will mostly move out of our cell. And since our cell will shrink, this is a hypertonic solution. Okay, what I would like for you guys to try and do is pause the video and see if you guys can fill out this example right here. And then after you finish, play and then check your work. All right, hopefully you guys took some time to do this next example. So first thing I'm gonna do, check out the amount of water. I can see that there's more water outside. So if this happens, my water is going to mostly move in. If I have water entering my cell, it's going to be like a hippo. So this is a hypotonic solution. All right, let's do two more. Same thing like you just did. Pause the video, do the examples, then check your work. All right, first thing I'm going to do, see if I could have the amount of water. I have 70% of water outside of my cell. Inside of my cell, it says I have 25% sugar. So if I subtract 100 minus 25, that means I have 75% water. So if I'm filling this out, water would be equal. And remember, just because it's equal, water is still going to move in and out. And if we're already at equilibrium, this is called an isotonic solution. All right, last example. 30% sugar. If I subtract 100 minus 30, I have 70% water. And then inside my cell, I have salt and sugar. So my combined amount of solute would be 35%. So 100 minus 35 would give me 65% water. Okay, so I would have more water outside. Water is going to move in. Water moves in. My cell will get bigger, like a hippo. So my solution is hypotonic. All right, so osmosis in cells, like we said a little bit earlier, if the concentration gradient or the difference in concentrations is too big, sometimes your cells can be destroyed, so they could burst.